Welcome to the first seminar of the year by Liz. Liz is Jacob Hameson and Beverly Mecklenburg lecturer at School of Physics at University of Melbourne, where she did her PhD, and then she went on to UC Irvine in the lab of Enrico Grattan, who is an expert on FCS, film, and all those things. Post that, Liz came back to Australia in the lab of Kat Gauss at UNSW, where she started applying these techniques to nuclear architecture, and she's been pursuing and doing fantastic work on those fronts ever since. Along this journey, she's won a lot of accolades, including Young Researchers, Young Fluorescence Investigator Award at the Australian Society of Biophysics, and Macaulay Hope Prize for Original Biophysics. So with that, just a couple of instructions. If you have questions, you could ask at the end by using the raise hand function that you can find at the bottom, or you could type in your question and answer. You could type in your question in the Q&A section in the app. Right, Liz, the stage is yours. Take it away.
Okay. Yeah. So thank you to Sentil for the opportunity to in this series. So today I'm going to talk about quantitative imaging of nuclear architecture and DNA target search in a living cell. So essentially our group is interested in the fact that there's approximately 2 meters of DNA inside the nucleus of each cell. And given that the diameter of a human's nuclei is around 10 microns, we're interested in how DNA is folded and compacted into this volume and how proteins can navigate such a complicated structure to arrive at a target DNA sequence. And we're mainly interested in the dynamics of this organization. So not only how is it organized, but how does it rearrange as a function of time? Because it's been shown that local rearrangements in the folding of DNA around histones into what we call a chromatin fiber can modulate where proteins bind the DNA sequence, and in the context of transcription, for example, what genes are expressed. So we're interested in this from the point of view of the chromatin architecture, but also the proteins that are navigating this structural framework to arrive at a target DNA sequence. So to investigate this area, we use fluorescence microscopy, and in particular, we use live cell laser scanning confocal microscopy. And here's a time series of images of a HeLa cells expressing a protein tagged to GFP. And essentially in every pixel of an image, we can access information about the structure that exists there, as well as the proteins that are navigating that structural framework via something called fluorescence correlation spectroscopy, which is based on measuring the movement of fluorescent proteins through a small observation volume and studying the fluctuation in fluorescence intensity that results, because this can give us information on the protein mobility and concentration. You can also do this as a 2-channel acquisition and therefore probe complex formation if you have another protein of interest or another element of the structural framework labeled with a red fluorescent protein. And so you can measure binding interactions as well as complex formation. Also in every single point, you can do something called a moment-based brightness analysis, which can give you information about whether or not a protein is a monomer or self-associates into a tetramer. And then the other type of spectroscopy we perform is a fluorescence lifetime analysis. And this can give you information about, for example, FRET interaction between 2 fluorophores that are appropriately labeled. And so, for example, in the context of the nucleus, we've used this interaction, for example, to probe nanoscale rearrangements in chromatin structure, because FRET is a phenomenon that only occurs on a scale of 10 nanometers. And by taking advantage of this and probing the fluorescence lifetime of the fluorophores involved in this interaction, we can spatially map chromatin compaction at the level of nucleosome proximity. So today I'm going to describe 3 projects that really make use of these 2 types of analysis, fluorescence correlation spectroscopy and moment-based brightness analysis, which collectively you could call fluorescence fluctuation spectroscopy. And specifically, I'll first give an overview of quite an old story now, but it basically gives you the background required to understand fluorescence fluctuation analysis, which is this type of spectroscopy that enables you to look at the diffusion of molecules and indirectly probe the accessibility of the nuclear landscape. And then I will talk about two projects that look at biologically active proteins and how you can use fluorescence fluctuation spectroscopy to track, for example, DNA repair factor recruitment to a DNA double-strand break, or in the context of transcription, how you can image transcription factor complex formation and look at how transcription factors use self-association to modulate their DNA target search strategy. So first is this idea of looking at the diffusion of an inert protein, such as EGFP, as a means to indirectly probe how molecules navigate nuclear architecture. So I've mentioned fluorescence fluctuation spectroscopy, but I thought I'd give a brief outline for people who are not familiar with it. It's essentially the analysis of fluctuations in fluorescence intensity due to fluorescent molecules moving in and out of a small observation volume, which in the context of a confocal microscope is the point spread function of the laser focused at the objective. So you can see here that if you have a confocal volume and a molecule diffusing throughout this small observation volume, that's about a femtoliter. Every time a molecule diffuses through here, there will be a fluctuation in the fluorescence intensity as it enters and exits this observation volume. And if we follow those fluctuations as a function of time, we find that there's a lot of information contained within this time trace. In particular, if we look at the amplitude changes and the duration of these fluctuations, we can get information about how the molecules at that location in the cell are moving, how many molecules are there, and what is their brightness. So here we have an example of a fluorescence fluctuation, and you can analyze the amplitude to get how many molecules you have there and how bright they are, and the duration to essentially get how fast they are moving. And this analysis of the amplitude comes under what we call moment analysis, and the duration comes under what we call fluorescence correlation analysis. And fluorescence correlation essentially analyzes how similar is this fluctuation with respect to itself at increasing time delays. And by doing that, can get information on how fast the molecules are moving through this observation volume. And so if you fit this decay, for example, to a model of diffusion, you can get the diffusion coefficient. If you look at the amplitude of this decay, you can get how many molecules you have in that observation volume. And as I mentioned before, if you do something called a moment analysis, you can also get the oligomeric state. So fluorescence correlation spectroscopy and application of it to EGFP, which is an inert fluorescent tracer, has been used extensively to indirectly quantify the accessibility of chromatin structure, because this will give us an indication of what, for example, a fluorescently tagged transcription factor or DNA repair factor would experience when they're diffusing throughout the nucleus. And so a long time ago, there was a study where they labeled the the chromatin network with RFP labeled H2A, which is a component of the nucleosome, and asked the question if they co-expressed EGFP and did a fluorescence correlation analysis of EGFP fluctuations in fluorescence intensity at locations where the chromatin, as revealed by the RFP signal, was quite compact or decompact. Does it have an impact on how an inert protein diffuses at that location? And they did this at various points throughout the nucleus and derived a map of EGFP diffusion. And surprisingly, they found that the local diffusion coefficient was independent of the local chromatin compaction status. So it didn't matter if the chromatin was high intensity according to RFP or not, the EGFP molecules exhibited a similar local diffusion coefficient. So in response to this study, we asked the question, maybe there's a spatial dependence on the diffusion with respect to local chromatin structure. So while EGFP could diffuse as fast in euchromatin versus a heterochromatin region, maybe the diffusive route it adopts with respect to this structure is modulated. So rather than doing these single point FCS measurements where you park your laser in a specific location in the nucleus and acquire the intensity as a function of time, we acquired a line scan across the chromatin network and spatially correlated these fluctuations in fluorescence intensity that were acquired as a function of time. And by introducing a spatial component into the analysis, we were able to ask the question, is EGFP forced to diffuse around this structure, although there may be some EGFP molecules in there diffusing? Or can EGFP diffuse throughout other structures? And if so, is there a delay to the arrival time in the structure versus getting out? So by transitioning to a line scan, we're able to see is there a spatial dependence on how molecules in the nucleus traverse this structural framework. And we use something called pair correlation analysis. And this is the type of fluctuation spectroscopy that the majority of my talk today will be focused on. So I'll just give you an overview of how it works. You require a confocal line scan as a function of time, and you get this chymograph, which reports how the molecules along your line scan are moving. And for example, if this line was acquired across EGFP molecules diffusing throughout the nucleus, and I was interested with the EGFP molecules at this location ever diffuse to this location, and if so, how long do they take? I could correlate the intensity fluctuation acquired at this location with an intensity fluctuation located at this one for all possible delay times. And I could get a decay that would tell me about how long the molecules on average take to arrive at that second location. And because it's a line scan and I have pairs of points along the line scan, I don't have to do it just for one pair. I can do it for all pairs of pixels along my line scan and essentially map molecular flow along my line scan. So here I can see the molecules arrived on a fast time scale. At this position, the molecules don't diffuse to the second location, so you get this anti-correlation. Here I get this arc feature where the decay is now on a delayed time scale, and this would suggest that the molecules encountered a barrier to diffusion. And so using this idea, we were able to look at how does heterochromatin or compact chromatin versus open chromatin regulate EGFP diffusion in the nucleus. So for example, if GFP was forced to diffuse around a structure versus being able to move through it, if we simulate these two scenarios, you can see in the pair analysis that they have a very distinct pair correlation carpet. So here you have diffusion outside this structure and then a lack of correlation to enter on either side of the structure. Whilst here, if the chromatin is accessible, but on a delayed timescale, you get this double arc feature as EGFP enters and exits this environment, and you can read off the time in milliseconds for the entry and exit. So using this idea, we then probed how the chromatin network regulates EGFP diffusion throughout the nucleus. So here we have a HeLa cell expressing free GFP, and we've stained the chromatin with Herx 3342. And if we scan a line across a heterogeneous region of DNA density in the EGFP channel, you can see that EGFP accumulates in some locations and is excluded from others. So if we analyze how does EGFP move along this line at a distance of 8 pixels, so it's a 64 pixel line, we're asking the question based on this Herx intensity profile, can EGFP diffuse from this low DNA density region into this high DNA density region? And can EGFP molecules in a high DNA density region leave this and go to the low DNA density region? And we find that on the timescale of our experiment that the GFP molecules can't perform this transit. So there's no communication in terms of GFP diffusion between low and high DNA density. If we then change our analysis, so we now do a pair correlation at a larger distance and say, can EGFP diffuse around a high DNA density region? Or if EGFP is in a high DNA density region, can it diffuse throughout this structural framework to another point of high DNA density? And we find that in this scenario that indeed the GFP molecules can perform these two transits and on different timescales. So collectively, this analysis was able to tell us that EGFP can't diffuse from low to high DNA density, but it can diffuse around high density DNA environments on a timescale of 2 milliseconds. And going from high to high, importantly, is also possible on a longer timescale of 8 milliseconds. And so collectively, this suggests that the chromatin network is behaving like a channel of high DNA density regions and low DNA density regions, but there's no communication in between on the timescale of our experiment. So this was interesting, but it then begged the question, if they're in both environments, how do they get there in the first place? And we investigated this further by cutting up this line scan into temporal segments and then testing, okay, so maybe GFP molecules don't get into the high DNA density regions on the timescale of the experiment, but maybe it's a rare event. And we found that indeed, if you cut the intensity carpet into temporal segments, you see moments of where communication is possible from a low to high DNA density region or a high to low DNA density region. And these bursts of communication are statistically rare. And so this is why we didn't see it in the previous analysis. So this channeled molecular flow, we found it to be ATP dependent. So if you deplete the cells of ATP, you reduce these bursts of communication between the low and high DNA density regions. But most importantly, what it taught us is that the chromatin network regulates diffusion in the nucleus, and this baseline behavior would have important implications for biological proteins such as transcription factors or DNA repair factors. And we also showed that it's cell cycle dependent. It's maintained in different animal models such as C. elegans and the Arabidopsis root. And so after performing a series of studies looking at how EGFP diffuses in these different biological systems, we then wanted to go on and look at how do DNA repair factors tagged to GFP or a transcription factor tagged to GFP navigate the structural framework of the nucleus. So this brings me to the next two talks, which are now focused on biological proteins. But the additional requirement of a biological protein is in some way you need to be able to track the active form of that protein. So there'll be a population diffusing throughout the nucleus like GFP not interacting with the DNA network. But then how do you differentiate that population from the active population that are actually doing something and interacting with the DNA template? And so I'm going to go into two different proteins that employ oligomerization when they're active, and this gives us a way to actually track that active population. So in the context of DNA repair, diffusion is also the main mode of transport for DNA repair factors, and they're constantly scanning the genome looking for genomic lesions. And when DNA damage is detected, the DNA damage response will signal that lesion's presence and recruit DNA repair machinery to that genomic location. And the consensus is that nuclear architecture is in some way actively facilitating this process. So there's been opening events at the double-strand break site detected, as well as compacting events that collectively facilitate repair factor accumulation at those locations. But an outstanding question is, how do DNA repair factors arrive at a double-strand break that can occur anywhere in the genome? So one DNA repair factor that we're interested in is 53BP1, and it's critical for repair of double-strand breaks, and in particular the repair pathway choice. And 53BP1 dimers are what are thought to be the active form, and they're known to recognize the unique histone code associated with the double-strand break. But how these dimers are actually biophysically recruited to the double-strand break prior to recognition of this unique histone code remains unknown. So here's a schematic of a nucleosome, and what's known with respect to 53BP1 is that it must bind to H4K20 methylation, and it also must recognize H2A15 ubiquitination. Size exclusion chromatography has also shown that 53BP1 dimers are the form that actively bind these to the nucleosome in vitro, but it's never been shown in a living cell whether or not the 53BP1 monomer is recruited, recognizes these histone marks, and then a second subunit arrives and they form a dimer at that point. Or does the 53BP1 dimer pre-assemble and then arrive as a dimer and interact with these two histone marks? So we wanted to investigate the spatiotemporal dynamics of 53BP1 dimer recruitment to these double-strand break sites that can occur anywhere within the genome. And to do this, we use fluorescence fluctuation spectroscopy. So we use this pair correlation analysis that I've just described, but we also couple it with something called moment analysis, which is also fluorescence fluctuation spectroscopy, that enables us to identify where the 50BP1 dimers are versus monomers. And we coupled this with a genetically inducible double-strand break cell system that enables us to recruit, to cut double-strand breaks throughout the nuclei and then track 53BP1 recruitment to those locations. So here's the genetically double-strand break inducible system. It's called DIVA, and essentially what happens is you have this U2OS cell line, which is a human cell line, and we can label how many double-strand break sites there are endogenously by immunofluorescence against H2AX. You can see that once you add this restriction enzyme that cuts double-strand breaks at approximately 100 locations throughout the genome, you see that these sites are illuminated by the H2AX immunofluorescence. And we found that within 1 hour of addition of this restriction enzyme, you can start to see the double-strand break sites within this U2S cell line. So this was useful, but in the case of our study that was wanting to be performed in live cells, we needed a way to label where are these double-strand break sites occurring. And given that H2AX immunofluorescence is limited to fixed cells, we first checked if we use 53BP1 accumulation, does it accurately report where the double-strand break sites are occurring? We found that there is significant co-localization between H2AX and 53BP1. And indeed, we could use the 53BP1 intensity as a map of where the double-strand breaks are being induced. And 53BP1 is also the DNA repair factor we're interested in terms of its at these sites. So fluorescence fluctuation spectroscopy, I've talked a bit about pair correlation analysis, but this other mode, moment-based brightness analysis, is what's going to enable us to first investigate where is 53BP1 dimeric versus monomeric, and is it indeed the dimeric form that we need to track by pair correlation analysis in order to understand how this repair factor is recruited to a double-strand break site. So in the case of moment analysis, again, it involves acquiring a time series of intensity images. In this case, you can do images. It's a lot of fluctuation spectroscopy can be a single point, a line scan, or images. But in the case of this specific experiment, we acquired a time series of images, such that in every pixel, we have our observation volume and we have a fluctuation in fluorescence intensity at that observation volume. So if we have a time series of images of 53BP1 tagged to GFP, in every pixel, we have a fluctuation due to GFP tagged 53BP1 molecules diffusing in and out of that observation volume. And if 53BP1 is a dimer at a location versus monomeric at a location, while they might have the same average intensity at those two pixel positions, what will be different is the variance in the signal. So if 53BP1 is a dimer at a location, there'll be a greater variance in the fluctuation fluorescence intensity at that location versus a pixel where 53PP1 is a monomer. And by using this principle, we can extract the brightness at each location. And if we know the brightness of our monomer, which is EGFP, we can extrapolate what we'd expect to be a dimer of EGFP or a dimer of 53BP1 in the case that it's labeled with EGFP. So the first moment is the mean. So this is the average intensity. And the second moment describes the variance of this signal, which is giving us information on how bright are the molecules at that location. And by taking the ratio of these two variables, we obtain the brightness in each pixel of our time series of intensity images, which is going to be our readout of 53BP1's oligomeric state. So I mentioned that in order for this method to work, we need to know what is the brightness of our monomer, which would be EGFP in this case. So we take a time series of intensity images in a cell expressing free GFP, and we calculate the brightness in each pixel, which gives rise to this distribution. And we can set up a series of cursors that highlight the monomer of EGFP. And based on this monomeric brightness, we can extrapolate what we'd expect a dimer of EGFP or an oligomer of EGFP to exhibit if it was formed in the presence of, when tagged to 53BP1. So here we have our EGFP control, and more or less everything is monomeric. If we then perform the same experiment in the cells now expressing EGFP 53BP1, you can see here we have a wild type 53BP1, the region of interest. Here we have a dimer mutant of 53BP1 to check that our cursors are right and that we should really see only monomers of 53BP1. And here we have a constitutive oligomer mutant of 53BP1 where we should promote oligomerization. And when we do the number and brightness analysis or the moment-based analysis of these three experiments, you can see from the brightness histogram that we've entered into the higher cursors that contain dimers and in some cases higher order oligomers. And if we quantify the number of pixels in these three experiments that contain dimer, you can see the wild type 53BP1 has a significant dimer population. The mutation that inhibits dimerization inhibits this dimer population, and the mutation that promotes oligomerization increases the dimer fraction. And we can also spatially map this throughout the region of interest using these cursors. So teal represents monomer, light green dimer, and red pixels higher order oligomer. So now we've confirmed that 53BP1 is indeed present as a dimer throughout the nucleus, and our dimer mutant indeed inhibits this dimerization, and we can quantify this across multiple cells, and we can confirm this by a HOMO-FRET experiment. So now that we know that 53BP1 is present at a dimer inside the nucleus, independent of double-strand break induction, the next thing we wanted to do is see what happens to this dimer population when we do induce a double-strand break, because we're interested in understanding how 53BP1 arrives at a double-strand break. So here we show again that the 53BP1 overlaps with where the double-strand breaks are being induced. And here's a time series of the DIVA cells before and at different time points after addition of the restriction enzyme that induces double-strand breaks throughout the nucleus. And so you can see that the intensity, that there are points where the 53BP1 starts to accumulate. So these are the intensity images, and these are the corresponding brightness maps that report 53BP1 oligomerization at these different time points. And they're pseudo-colored according to monomer, dimer, and higher order oligomer. And you can see that before we've induced the double-strand breaks with addition of the restriction enzyme, there's a significant population of dimer throughout the nucleoplasm. But after we induce double-strand breaks, this dimer population starts to redistribute and be lost from the nucleoplasm as more double-strand breaks are induced and a higher order oligomer accumulates at those locations. So this suggests that the first instance that the 53BP1 dimers are, like reported in vitro, the population that are being recruited to double-strand break sites. And this is important for how we approach the pair correlation analysis. But to confirm this, we perform this analysis across multiple cells, and via a mask analysis, we're able to demonstrate that there is a significant reduction in 53BP1 dimer from the nucleoplasm as a function of time. And while the composition of the 53BP1 foci does not change as a function of time, it's the number of foci that does change. So when you lose the 53BP1 dimers from the nucleoplasm, as detected by the brightness analysis and verified by HOMO-FRET, you see an increase in the number of foci that are formed, and at these foci, you see an increase in oligomer formation. So collectively, this suggested to us that the 53BP1 dimers are extracted from the nucleoplasm, redistributed to these double-strand break locations. And at those locations, they accumulate and potentially assemble into higher order oligomers. So how do the 53BP1 dimers accumulate at these locations? So this comes back to our original question: how is 53BP1 biophysically recruited to these sites? So to investigate this, we used the pair correlation analysis, but we used a slightly different version to what I previously presented. In particular, we use something called cross-pair correlation analysis. So now that we know it's the 53BP1 dimer population that's been recruited to the double-strand break site, this is the population we wanted to track in order to understand how this repair factor is biophysically recruited to these locations. So we labelled 53BP1 with EGFP and MK2 and acquired a line scan across a double-strand break location in both channels. So what this enables us to do is not only analyse fluctuations of recording 53BP1 movement along a line scan that transects through a double-strand break foci in both the EGFP and the MK channel, which it also enables us to do what is called a cross-pair correlation analysis. And in this instance, track when does a 53BP1 molecule tagged with EGFP move with the exact same dynamics as a molecule tagged with MK. And under these conditions, you would expect the only way that would be possible is if 53BP1 is diffusing as a dimer. So now we have a way to look at the dynamics of 53BP1 monomers and dimers moving onto a double-strand break site. But we can do a cross-pair correlation analysis and extract the dynamics of only those heterodimers where 53BP1 is tagged MK and EGFP. So here we have an example of the U2S DIVA cell expressing 53BP1 EGFP as well as 53BP1 MKs. And we trace a line scan across a foci in both channels. So here's the foci, and this enables us to have intensity fluctuations that we can later cross-correlate. And we tested different distances, and essentially what we found is that if you go from PCF 4, 7, and 12, you can see that you get this fast arrival time, then it gets slower, and then you lose the molecules. It's more clear in the MCAPE channel. So they arrive on a fast time scale, then the delay is shifted along this log axis, and then you lose the molecules. And this is also true in the cross-pair correlation analysis. So collectively, what this tells us is you can track the molecules at PCF7, but if you go further than this, using 12 pixels, you've already lost the molecules. And the advantage of PCF7 along a 64 pixel line is that we can ask the question, how long does it take 53 BP1 to diffuse from the nucleoplasm onto a DSP foci? And how long does it take 53 BP1 molecules to diffuse off a DSP foci back into the nucleoplasm? So we now have a way to quantify 53 BP1 recruitment versus retention. And by doing the cross-pair correlation analysis, we can pull out specifically the 53 BP1 dimer population. And what we found is that when you do this at a distance of 7 pixels, you find that if there is no foci, actually you can't detect 53 BP1 movement on this spatiotemporal scale. But only when a foci is present, as shown here, you see a large population of molecules diffuse onto this structure on a fast time scale, or the limit of our line scan actually. So they all arrive on a time scale that exists from a millisecond time scale through to seconds. But in terms of the population getting off this DSP foci, you don't ever see 53 BP1 molecules getting off this structure on the time scale of our experiment. So this tells us that indeed 53 BP1 dimers are recruited onto DSPs, which is in agreement with the brightness analysis. And due to a significant difference in the on versus off rate, they're able to accumulate at these locations. So now that we had a way to detect how 53 BP1 molecules are recruited to these locations, we wanted to investigate the impact that the histone code unique to a double-strand break site has on the retention, or the recruitment versus the retention at these locations. So now we have 53 BP1 mutants that don't recognize the H4K20 methylation mark versus the H2A K15 ubiquitination mark. And when we do the brightness analysis in the wild type setting, remember we see that there's a 53 BP1 dimer population located throughout the nucleoplasm, as highlighted by these light green pixels. And after double-strand break induction, this population redistributes to double-strand break sites, leaving behind a majority monomeric population. But in the case of the H4K20 methylation mutant, what happens is there's a dimer population, but after you induce double-strand breaks, actually this population doesn't redistribute to sites where the double-strand breaks were presumably cut. And in the case of the H2A K15 ubiquitination mutant, again, you have dimers still being formed prior to double-strand break induction, but you have some recruitment to the double-strand break sites, but importantly, not this higher order oligomer formation. So there's a difference in the way the dimer population is responding to the double-strand breaks, and it's dependent on this history. So to further dissect this, we did the cross-pair correlation analysis. So in the wild type setting where we see significant recruitment of 53 BP1 dimers to these locations, you can see this from quantitation across multiple cells. We saw that this accumulation results because the 53 BP1 dimers can arrive on a double-strand break site, but they can't get off this structure on the timescale of our experiment. If we look at the mutant that inhibits recognition of H4K20 methylation, you see that there is no significant recruitment of the dimers to these locations. And if we identify the odd foci based on 53 BP1 intensity, not dimer localization, and we do a line scan analysis across that foci, we find the reason there isn't a redistribution of the dimeric population to these locations is because while they can arrive at these locations, there's a significant fraction that can get off the structure. And so this must lead to the lack of accumulation at these locations. And interestingly, in contrast to this mutation, if we look at the H2A K15 ubiquitination mutant, where again, we actually don't see significant relocation from this nucleoplasm to the foci. The reason we potentially don't see foci formation at these locations, oligomer formation at the double-strand break foci locations, is that there's a significant loss in the efficiency of molecules that arrive there, although they can be retained there. So this was interesting because this histone mark seems to be important for retention, while this histone mark seems to be important for recruitment. And so there's a differential role in the histone code at a double-strand break site in maintaining significant 53BP1 retention at these nucleic locations. So in conclusion, from coupling the double-strand break inducible cell system, BEVA, with fluorescence fluctuation spectroscopy, we were able to study 53BP1 spatiotemporal dynamics. And in particular, we found that 53BP1 dimers could be tracked by extending the pair correlation to a cross-pair correlation analysis. And we could track the redistribution of this dimeric fraction in the nucleoplasm to the double-strand break foci. And we find that there's a pre-existing pool of 53BP1 dimers and that they do bind as a dimer to this structure. And the different histone marks at this structure have a different role in terms of retaining 53BP1 dimers at this location. In particular, we saw that the H4K20 methylation mark is important for retention, while the H2AK15 ubiquitination mark is important for the recruitment to these locations in the first place. And so now what we want to do is look into the role of chromatin architecture in recruiting 53BP1 to these locations and measure via this FLUORESCENCE assay that I don't have time to talk about today, how chromatin architecture participates in enabling 53BP1 dimers to accumulate at these locations. So this cross-pair correlation analysis was very useful in the context of 53BP1 because it's the dimer that is the functional version of 53BP1 that we wanted to track. But in the case of transcription factors, they not only form dimers, they also form higher order oligomers, and cross-pair correlation analysis can't differentiate a dimer from a higher order oligomer. It will just see it as there's a positive cross correlation because a complex has been formed, and it can't comment really on anything more than that. So to address this issue, I was just going to mention as the last topic of this talk, a new method where we're looking at pair correlation of brightness fluctuations. So transcription factors regulate gene expression by site-specific binding to DNA. And from single particle tracking experiments, it's known that, again, diffusion is the main mode of transport, and it's coupled with non-specific binding events and then a specific binding event upon arriving at a target DNA sequence. But the thing is that transcription factors do not act alone. They're often regulated by complex formation and self-association. So while a single particle tracking experiment has very good spatial resolution and sensitivity in terms of how the molecules are navigating nuclear architecture, because you are tracking single molecules, it's hard to know the impact that complex formation has on that transit. And so if you go back to a normal fluorescent labeling strategy, like with EGFP, it's hard to then track the diffusive route these molecules take because everything is fluorescent. So we thought to take advantage of the fact that transcription factors employ oligomerization to regulate their DNA search patterns, because we have this brightness analysis that enables us to essentially differentiate fluorescently labeled transcription factors in their monomeric versus dimeric or oligomeric form. And so, for example, STAT3 is a transcription factor that is known to form dimers in the cytoplasm that are subsequently recruited to the nucleus, and depending on the DNA sequence, can further dimerize into tetramers. And so we thought that if everything's labeled GFP, sure, you can't see this process, how the dimers get into the nucleus and how the further dimerization event modulates the binding affinity for DNA sequences. But if we were to analyze the brightness of these different species, this would in some way give us a way to track these individual complexes, and similar to single particle tracking, give us a way to isolate a few molecules in a sea of monomers. So in the end, the monomer becomes our background because these brighter species become visible via the brightness analysis. So we wanted to develop a method that could track molecular mobility as a function of oligomeric state, and therefore map the impact live cell nuclear architecture has on transcription factor DNA target search. So I've already introduced this idea of brightness analysis, where in every pixel of a confocal time series of images, for example, you can ask the question, how does the intensity change as a function of time? Because we know that bright molecules will give rise to a big fluctuation, dim molecules, not so much fluctuation, and while they have the same average intensity, the ratio will be different, so we can differentiate these two scenarios. So this gives us information on the average of our oligomeric transcription factor, for example, but it doesn't really tell us about any heterogeneity in a single point. And so that's why we do tend to do these frame scan acquisitions, because we can then look at heterogeneity spatially. So for example, here we have a HeLa cell expressing GFP multimers. There's a monomer, dimer, and a pentamer. And you can see these three different species in the brightness analysis. And then you can go back and spatially map where they're located. But if I looked at that in a single point, it would be hard to detect these three species. So I have a way to investigate heterogeneity in terms of the oligomeric complex by doing a spatial analysis, or I can do a more sophisticated brightness analysis in a single point where I'm enabling high statistics. But the problem with both of these approaches is that neither really comments on how these different species are moving. So I can either have information on how they're spatially distributed, how heterogeneous they are in a single point, but in the context of transcription factors, neither really tells me what happens after that snapshot, and does the oligomeric form modulate the mobility of the transcription factor versus the dimer. And this is what we're ultimately interested in, like we were with 53BP1. What happens to the dimeric versus the higher order oligomeric form of our biological protein? So to investigate this, we coupled the pair correlation analysis with brightness analysis. So just to reiterate, if we did a line scan, for example, and we scanned this line rapidly as a function of time, and we recorded the fluctuations in fluorescence intensity of our transcription factor tagged with GFP, then we could correlate the fluorescent fluctuations in two locations and say there's a heterogeneous distribution of oligomeric states present. We would see those molecules arrive at another location, but we wouldn't necessarily know what oligomeric states gave rise to that arrival time. We just know that there was a number of molecules that arrived at the second location. So the idea is maybe we could correlate brightness fluctuations and sort the arrival time based on the brightness of the species. So instead of doing a pair correlation on intensity fluctuations, we first make these intensity fluctuations brightness fluctuations. We then do a spatial correlation of these brightness fluctuations. And now, rather than just counting how many molecules arrive at the second location, we start to filter them based on their brightness and look at how their oligomeric state modulates their arrival time. And so the idea is based on the amplitude of the arrival time. If you use a calibration similar to what we did for the brightness analysis for free GFP, we can then assign which species is arriving based on the amplitude of the correlation, and the peak time tells us the arrival time. So now if we do a line scan across a cell that's expressing a monomer, dimer, and pentamer of GFP, for example, and we look at how do these different species cross the nuclear envelope, we can see that you now can see the different peaks for the arrival time to get from the cytoplasm to the nucleus. And based on a calibration with the monomeric EGFP shown here, we can assign these to the dimeric or pentameric complex that we've transfected in. So now we have a way to really look at not only how does a dimerization event modulate the mobility of our biological protein, but how does an oligomerization event? So we applied this to STAT3 to ask the question, when STAT3 does form or undergo a dimer to tetramer transition, is it the tetramer that searches for this DNA sequence, or does the dimer then interact with another dimer after binding DNA and form a tetramer? So to investigate these two possibilities, we transfected cells with STAT3 mCherry as well as a DNA binding mutant STAT3 tagged with GFP. And you can see from the brightness analysis that we significantly inhibit dimerization in the DNA binding mutant case. So dimers are light green, monomers are dark green, and the oligomers are red. We then scan a line across the nuclear envelope and transform them into brightness fluctuations. And then we perform the spatial pair correlation of these brightness fluctuations for all pairs of pixels along the line scan. And we do this in both channels. And what happens if we calibrate the amplitude of the correlation in each pixel is we arrive at these carpets that are quite difficult to interpret, but I will extract the information that's important from this experiment and show you that if you choose a column that tests transits across the nuclear envelope, like how does STAT3 enter the nucleus, you can see that in the wild type case, STAT3 dimers arrive on a timescale of 300 and 500 milliseconds. But in the case of the DNA binding mutant, they can also arrive in the nucleus on this timescale as a dimer based on the amplitude of this correlation function. If we then look inside the nucleus at a column where we see dimers have formed, we see that in the case of wild type STAT3, there's a mobile population and an immobilized population. But in the case of the DNA binding mutant, there's only this mobile population. So we can see that DNA binding is important for immobilization of the STAT3 dimer. If we then look at another nucleic location, we see oligomer formed based on the amplitude of the correlation function here. We see that the STAT3 tetramer is immobilized and the DNA binding mutant never forms an oligomer. It just remains as this highly mobile dimer. So collectively, from doing this analysis, we're able to differentiate these two scenarios and see that the STAT3 dimers must actually bind DNA in order to form a higher order oligomer based on the mobility of these different species across the line scan. And we showed that actually the tetramer is related to transcription, because if we activate transcription with the cytokine that actually promotes STAT3 transcription, we see more of these tetramers being formed. So I just wanted to finish off by saying that the pair correlation of the brightness analysis along a one-dimensional line scan is useful, but chromatin organization is spatially complex. So it's useful for looking at how STAT3, for example, got into the nucleus, because you can draw a line across the nuclear envelope. But if you want to look at how it's navigating nuclear architecture, we really need a two-dimensional acquisition. And this is problematic just because you need a fast acquisition, but also from a visualization point of view. So now what we're doing is borrowing an idea from Malacrida and Hinde et al., where they do a pair correlation in this radial fashion from each pixel within a frame scan acquisition, for example. And so then you can look at, is your transcription factor diffusing out from this point isotropically or anisotropically? And so by mapping this in each pixel of a frame scan acquisition, we can start to ask the question, do the molecules follow a particular path and form these connectivity maps? So I'll just go over this briefly, but essentially now we're interested in not only looking at the arrival time, but how do these different species navigate DNA density? So we use the brightness analysis, but we now do it in a frame scan acquisition. And so we transform our intensity movie into a brightness movie. We filter it out into the dynamics of the monomer, dimer, and oligomer. And then we do the radial pair correlation on every pixel within each channel. And this gives us not only the arrival time in terms of a two-dimensional environment, but it also enables us to map the anisotropy of this motion. So where is it interacting with an obstacle? And we found from preliminary data applying this to STAT3, for example, that I won't go through the simulations. Just go straight to the STAT3. That actually you can detect the different arrival times. So here we have STAT3 accumulating in the nucleus. We acquire our time series of intensity images as a function of time. And we see that there's a nuclear accumulation of this transcription factor. So if we look at the dynamics of this protein before stimulation versus after, you can see there's a massive redistribution from the cytoplasm to the nucleus. And if we take snapshots out of our time series of intensity frames and just derive a brightness map, you can see that the dimers in light green move into the nucleus, leaving behind a monomeric population. And inside the nucleus form this tetramer that's shown here in red. And so if you derive a connectivity map or a map of anisotropy for how these different species are moving prior to STAT3 stimulation versus after, you can see that in the nucleus you have this higher order oligomer being formed and that it experiences obstacles that are different from the dimeric form of this transcription factor. And we can go into this region of interest at time points that are relevant to how STAT3 was shown to be recruited to the nucleus from the lines data and see that the dimer is moving on a delayed timescale prior to stimulation. Then after stimulation, 15 minutes in, you see the dimer is moving quite fast now, so it's sped up and an oligomer is formed on a delayed timescale. And then at the point where we see the puncture being formed, you see that the dimer goes back to being slow and the tetramer is even further slowed down. So we can track now how these different species are being recruited to the nucleus and navigating the nuclear architecture much more broadly than we could with the line scan. So in conclusion, the pair correlation analysis is it's a powerful way to track the accessibility of the nuclear landscape, and when it's applied to an inert molecule, you can really look at chromatin opening and compacting events. But if you apply pair correlation to an intensity information, you don't really have any information on how a biological protein might oligomerize. And we find from pair correlation of brightness fluctuations that you can now look at how dimer versus tetramer formation affects the mobility of your fluorescent protein, because this method amplifies the brighter species and then filters the dynamics based on the arrival time. And so the future for this method is, well, we have to publish the two-dimensional version, but now what we're doing is cross-pair correlation of molecular brightness analysis and trying to identify which of these species are interacting with RNA pol 2, for example, or other STAT transcription factors in this family. So with that, I just want to acknowledge the people that are involved, in particular Jae-Chong Liu, who led the first project on 53BP1 oligomerization, and Ashley Solano, shown here, who has led this project on the pair correlation of molecular brightness. And the other people in our group, our collaborators, Enrico Lorenzo, Gail and Katerina Gauss, as well as Sentil for organising this seminar, and for your attention.
Thanks, Liz. Great talk. The floor is open to questions. I think while people start gathering their questions, so let me ask something that I was curious about. So the oligomerization of these molecules, are they always existing in an equilibrium, or do they modulate that in response to some activation or whatever, stress in cells or whatever?
So in the case of 53BP1, it exists as a dimer under basal conditions throughout the nucleoplasm, and it doesn't, double-strand break induction doesn't seem to change the number of dimers are present, it just redistributes them in the nucleus. Whilst in the case of STAT3, the 53BP1 dimers are also redistributed, but it promotes self-assembly into higher order oligomers. But I guess the only population that we wouldn't be observing is if there's like an oligomeric population that's immobile. We will not detect that. And in the case of number and brightness, it's not sensitive to, it always reports the average brightness in each pixel. So if there's only a few dimers present, it won't be sensitive towards that because you're getting the monomers bringing that brightness down, whilst the pair correlation of molecular brightness will selectively enhance those species. So there's advantages in general. Cross-pair correlation of intensity fluctuations is best for observing a monomer to dimer transition, and the pair correlation of brightness fluctuations is not very sensitive. It can't distinguish monomers and dimers very well, but it's good at looking dimers to higher order oligomers.
Mm-hmm. Cool. There's a question by Ken Davidovich. I'm gonna unmute him so you can ask his question.
Yeah. So can you comment about the spatial resolution? How many nucleosomes you have per pixel in this FFS analysis?
Yeah. So all the fluctuation-based analyses are diffraction limited. In terms of the line scan experiments, for example, they are 3 micron lines, and the pixel size is around 80 nanometers. But we're obviously diffraction limited, so we're measuring diffusion either within a point spread function that has a radial axis of 260 nanometers, but at the same time we are pulling out single molecule information in terms of how those molecules are moving throughout that observation volume. So from the point of view of FCS, it's diffraction limited, although you can observe single molecule information. From the FLUORESCENCE assay looking at nucleosome proximity, again, it's diffraction limited, but you're observing proximity at an ensemble level within that observation volume. So the FRET is reporting changes in nucleosome proximity that change between 1 to 10 nanometers, but there must be many nucleosomes. It's the average proximity change in a 260 nanometer volume. So you're still diffraction limited, but you're indirectly accessing information that's somehow super resolved using the FRET as a readout. So you don't really know how many nucleosomes are present in an observation volume. You just know their average proximity.
So can I, so the reason I'm asking this is it, do you know that you really see a facultative heterochromatin or euchromatin, or is it possible that in each pixel you actually have mixture of both of them and the data is just an average? Is there any way to tell?
So I didn't show it today, but we have been looking at the so I think in the past we were maybe a bit misleading to call it heterochrome euchromatin, because now we're actually looking at these FRET maps in the context of H2.1 alpha localization or it being knocked out. And we're definitely measuring an ensemble effect. So if you knock out HP1, for example, you will see that the average nucleosome spacing increases, although you cannot see that from the intensity image. So there's those FRET maps, they report an ensemble packaging of the nucleosomes within each pixel, but it's certainly not directly related to whether or not it's heterochromatin or euchromatin. And it's a three-dimensional volume. So as you say, there's the radial axis is 260 nanometers, but actually in the orthogonal direction, it's closer to 800 nanometers. So that's the volume that you're sampling. I'm not sure if that answers your question, but certainly from our heterochromatin experiments, it's not clear cut as to whether or not heterochromatin has a closer nucleosome spacing than versus euchromatin.
Ken?
Oh yeah, thank you. You did answer my question.
All right, Liz, I was also curious about the HOMO-FRET experiment that you described. Do you just do steady state anisotropy with different polarizers, or do you also do lifetime anisotropy there?
No, yeah, we did steady state. So we just had a polarization cube in the detection. And yeah, it was surprisingly, again, it was interesting comparing the brightness with the HOMO-FRET, because the brightness seems to, because it's only good at detecting a mobile population, the HOMO-FRET sometimes gives you more information on species that aren't captured by the brightness analysis.
Yeah, I was just curious about how does that compare to number and brightness if you correlate the two results?
Yeah, so they agreed in the case of dimer formation, but in terms of the oligomeric state, the HOMO-FRET is much more sensitive towards the oligomerization. And I think that's because in NMB, you really need, you sometimes suffer from photo bleaching potentially as they move. And then once you lost that subunit, the fluctuation changes. But in the HOMO-FRET, there was more sensitivity towards higher order oligomer species than the brightness.
Cool. And just as a selfish question, do you have that system coupled to lifetime, or do you just have steady state?
No, we have it to lifetime, yes.
Okay. All right, I'm gonna catch up with you on that later. Right, are there more questions? Right, looks like there are not a lot of other questions. Nobody's asking any questions here. So yeah, Liz, thank you very much again for this fantastic talk, and we'll look forward to where all this journey through the nucleus takes.
Thank you, Santa.
Yep, thank you very much.
Thank you.
Thank you everyone for attending. Bye.